Good afternoon, everybody. Thanks for listening to this presentation. I'm Dr. Al Jergens in Veterinary Clinical Sciences here at the College of Veterinary Medicine. I'm focusing this presentation on the role of the gut microbiome and metabolome in GI health and disease. And my objective of this presentation is to show you some of the unique services that my laboratory group can offer in the context of your biomedical research activities. So who am I? I'm a clinician scientist here at Iowa State University. I'm a board certified small animal internist. I'm also co-founder of a biotech startup called 3D Health Solutions with two other founding members, Drs. Karen Allenspa and John Moschel here at the College of Veterinary Medicine. I have a very strong applied research focus. That's based upon my advanced training in veterinary pathology. I have my MS degree here from Iowa State University, as well as an immunopathology PhD degree. Of interest, my PhD program was funded through a KO1 award through NIH, where the focus of my research work was development of the murine-defined microbiota model, also known as the ASF model. And I'm sure that you've heard some previous background and use of this model from Dr. Michael Wannenmüller. By the way, Dr. Wannenmüller was my major professor in my PhD program. If I had to characterize our research focus here, it would be on translational medicine. And so broad applications from spontaneous occurring animal disease models to human diseases. I have marked expertise in comparative gastroenterology, looking at a variety of animal models, principally the dog and mouse, but other models as well. I have extensive experience and expertise in the performance of clinical trials in dogs, cats, and rodents, and in the initiation and culmination of collaborative research activities. I'm well published, have a number of research abstracts and an H-index of 36, which is the highest in my department. And so simply, I have an interest in host microbiota interactions that modulate GI health and disease. In particular, I'm interested in animal models of chronic colitis. This is where we can use the ASF immunocompetent mouse model, as well as the fact I have expertise with other murine models, probably most commonly the IL-10 knockout mouse. We perform clinical trials in dogs with chronic enteropathies with a focus on select underlying diseases, spontaneous diseases such as inflammatory bowel disease, colorectal cancer, nutritional trials that influence spontaneous diseases, and then endocrinopathies such as diabetes mellitus. I'm also actively involved in ex vivo modeling studies using intestinal epithelial organoids. This is much of the early work that I did with John Moschel and Karen Allenspa. I'm not going to talk about that per se because I know that both Drs. Allenspa and Moschel will be delivering their own presentations in these areas. That said, our intestinal epithelial organoid work is largely focused on drug discovery and gene editing. Along with these underlying procedures, we also have an active and growing bioarchive comprised of biologic specimens from these animal models, as well as organoids that are frozen. Then finally, and most recently, I've engaged with collaborators looking at the role of intestinal permeability as it modulates host microbiota interactions. And I now collaborate with an investigator that has the ability to perform sophisticated Using chamber work to take a look at paracellular and transcellular transport, once again in diverse animal models. And so I wanted to give you some examples of some of the work our laboratory group does. Here's an example using the ASF mouse model where you can colonize these mice with a pathobiont. In this particular course of experiments, we're colonizing with a pathobiont called Helicobacter bilis. And what this organism does, it perturbs the resident microbiota. It stimulates and simulates ongoing chronic intestinal inflammation. These data show the host responses that occur following Helicobacter pylori colonization, where we see increased antibody responses, T-cell reactivity, and proliferation. Obviously, over time, we see progressive and more intense transmural inflammatory responses that occur. These organisms create microbial dysbiosis. They translocate into the ileal and colonic mucosa. And once again, other manifestations of host responses are going to be these altered gene patterns that we see, particularly those involving inflammation, fatty acid metabolism, and detoxification. So think of these as key and core housekeeper activities in response to this pathobiont colonization. Another example, this time in the transgenic mice, looking at IL-10 knockout mice, where we've investigated the potential causal association between microbial imbalance and intestinal inflammation. And here you can see as inflammation ensues because of the genetic deletion in the IL-10 gene promoting a pro-inflammatory intestinal environment, we see changes in the complexity, the diversity, and the richness of the intestinal microbiota, where we lose those beneficial organisms such as the firmicutes, the bacteroidetes, and we see a bloom in those potentially harmful organisms such as the Enterobacteriaceae. Again, a very nice traditional rodent model of intestinal inflammation. Next, I want to give you an example of some of the spontaneous diseases that we see on the clinic floor here at the teaching hospital. And so Dr. Allensbaugh and I are the two internists that see the bulk of the gastroenterology cases here. We have a vast and diverse, really strong referral caseload. And we see cases like this boxer that has chronic diarrhea. And we know, based upon previous studies, that there are certain canine breeds that have defects in their innate immune sensing, just like you and I do. Humans with IBD, either Crohn's or ulcerative colitis, collectively have roughly 240 mutations that put them at increased risk for chronic intestinal inflammation. Well, we have certain breeds that also occurs. Soft-coated Wheaten Terriers, Boxers, French Bulldogs, German Shepherd dogs would be some of the breeds. And so we work these up identically as would occur in you or I. And so we can perform full GI endoscopy on these animals. We don't have to sacrifice them. We can anesthetize them, go in there through an upper GI procedure, take a look at the upper portions of the bowel, and then turn these animals around following colonic preparation, and we're able to do full colonoscopy and ileoscopy. And I'm showing you an example of some of these lesions that we pick up in these animals. Nodular granularity, secondary to this really hyped up immune system. Why? Because of the intestinal dysbiosis, and then these mucosal erosions that we can see and biopsy directly in these animals. And over the past, I would say 15 years, we published a number of studies looking at the microbial imbalances that occur in dogs with chronic enteropathies, including inflammatory bowel disease. In this upper right, I show you a histogram where we looked at predominant bacterial taxa in dogs with IBD compared to healthy control animals, showing that these IBD dogs, again, suffer from marked microbial imbalances. Evidence of this is further shown in the lower right. This is 3-color fluorescence in situ hybridization, where you can see this intense and thick adherent mucus biofilm that occurs in these animals. On the upper left, I give you an example of the boxer that I just previously showed you that has a population of adherent invasive E. coli within its colonic mucosa. Why? Because it suffers from a genetic mutation in the NCF2 gene, just like occurs in humans, that allows its innate immune system to ineffectively clear these mucosal organisms. And so I want to emphasize to you that not only can we perform clinical studies, but we can apply advanced molecular techniques to investigate mechanisms behind chronic intestinal inflammation. This leads me to the next part of this presentation I wanted to talk to you a little bit about, which are therapeutic interventions by nature of microbial dysbiosis. And so we can address this problem in a number of ways with dietary trials, using a hydrolyzed ration. We can treat these animals with prebiotics or soluble fibers that generate short-chain fatty acids, normalize motility, bind irritants, including excessive amounts of bile acids. We've done a number of studies looking at probiotic interventions in dogs with chronic enteropathies, and I'll share more about that in the near future. And we haven't done a lot with fecal microbiota transplantation. The concept here is that it's supposed to alter the luminal microbiota and it may improve metabolic products. Now, what the human literature would suggest is FMT is extremely useful for C. difficile infection. However, the use of FMT for chronic intestinal disease, such as Crohn's disease and ulcerative colitis, is much less pronounced and much less definitive. And so Dr. Allensbaugh and I both feel that the type of dysbiosis that we see in dogs with chronic enteropathy, and again, I shared that with you, these marked microbial imbalances, these thick adherent biofilms, really this presence of a complex dysbiosis make FMT a less amenable treatment modality that we see in our dogs. Also the concern over passing on antimicrobial resistance. And so why all the interest in probiotics? Well, that's largely based upon the very positive data that we've seen in rodent models that are in the literature. And so be them transgenic rats, be them genetically engineered mice, we've had good results with particularly multi-strain probiotics with regards to inducing clinical remission and improving histologic lesions. When we look on the human side in the human literature, probiotics have been shown to be beneficial with regard to ulcerative colitis patients, not only in inducing remission, but also potentially in preventing relapse. We don't really know how probiotics work in you and I, and we don't really know how they work in dogs, but that's an area of active investigation that we have, and I'll share some of that data with you. So perhaps probiotics enhance local immune regulation through secretory IgA secretion. They may enhance epithelial barrier function. And so do they upregulate those tight junction proteins? I'll show you some compelling data that we've found in our clinical trials. Remember, probiotics also promote competitive exclusion. So we keep those pathogens and pathobionts out. And then finally, these organisms can produce their own antimicrobial substances called bacteriocins. So whether it's one mechanism or some combination of these mechanisms, that's an area of active investigation that we have not only in our canine model of spontaneous enterocolitis, but also in the notobotic mouse model that I've worked with Mike Wannenmuller on. And so just a brief table showing that there's not an extensive amount of evidence on the use of probiotics in dogs with chronic enteropathies. The red arrows will highlight the two largest studies that we've done here at Iowa State University and a multi-center approach with an Italian group, and I'd like to talk a little bit more about those. So this first study was a randomized open-label trial in 20 IBD dogs, and here we were looking at the effects of an 8-strain probiotic cocktail termed VSL3. And so this probiotic was given at phenomenally high doses, ranging from 112 to 225 billion, yes, billion with a B, CFU per 10 kilos of body weight. And so half these dogs were treated with VSL3. The other 10 dogs were treated with traditional chemotherapy consisting of an antimicrobial, metronidazole, along with an anti-inflammatory, prednisone. Outcomes included clinical improvement, histologic scores pre and post treatment. We specifically wanted to look at immune cell populations and tight junction protein expression. Then finally, we looked at the fecal microbiome using qPCR in these patients. So we'll cut right to table 3, which are the summary statistics for these two cohorts. And so the various outcomes are on the vertical axis, and then you can see the VSL3 group and the combination treatment group presented in front of you. T0 or baseline, that is pre-treatment, and then T1 is following 8 weeks of continuous therapy. And so all these animals, regardless of the therapy, improved histologically and improved clinically. That is, we were able to induce remission. However, those animals that received the multi-strain cocktail improved faster and to a greater extent. Most importantly, we looked at immune cell populations. Were they pro-inflammatory or were they down-regulatory? And what we saw is that at diagnosis, particularly in this VSL3 group, that they have an overt inflammatory profile, marked increase in the number of CD3 T cells following 8 weeks of VSL3 treatment. We saw marked attenuation of those pro-inflammatory CD3 cells, accompanied by an increase in down-regulatory cells expressing either FOXP3 or TGF-beta. So what we did is we dropped down that pro-inflammatory cell population, bumped up that down-regulatory population of those regulatory T cells that could promote mucosal homeostasis and reduce intestinal inflammation. Also, we looked at a select number of tight junction proteins in a small canine cohort, and what we saw in this particular study was that the tight junction protein occludin was significantly increased following probiotic administration. I wanted to show you that we can perform these studies easily. For me to do an upper and GI endoscopy in a dog takes roughly about an hour and 15 minutes. We obtain anywhere between 12 and 15 biopsy samples from 4 select organs: the stomach, duodenum, ileum, and multiple areas in the colon. And I've given you an example of those biopsies. And what can we do at these biopsies? We can do everything. And so we can not only take a look at tight junction protein expression, we can not only take a look at histopathologic lesions, but we can apply a multi-omics approach using these biopsy samples, taking a look at the microbiome and the mucosa, taking a look at the metabolome, taking a look at the transcriptome, and even the epigenome in these biologic samples. Well, that first study was great, and we focused on the fecal microbiota. Guess what? Even following the 8 weeks of probiotic administration, we saw no significant change in fecal microbial populations. So it makes sense to take a look at the mucosal microbiota. Remember, there's a difference between the luminal flora and what's going on either attaching or within the mucosa itself. So we performed a second study here at Iowa State. This was a randomized controlled trial, taking a look once again at 2 treatment arms. The first treatment arm was standard IBD therapy. And so these dogs received standard hydrolyzed ration and an anti-inflammatory in the context of prednisone at roughly 2 milligrams per kilogram at a tapering dose. The second arm was standard therapy, okay, so again, hydrolyzed diet plus prednisone plus the addition of the probiotic. Our outcome assessments were the same: clinical scores, histologic changes, tight junction protein expression, but instead of doing quantitative PCR, we looked at a 7-fish panel and took a look at individual mucosal bacterial populations using fluorescent in situ hybridization. And so the study design was baseline diagnostics and diagnosis of IBD, a recheck after 3 weeks, 5 more weeks on the probiotic, and then rescoping, reassessing these animals clinically and biologically at 8 weeks. So what does the data show? I put this cartoon up there to show you where we expect to see bacterial populations in the distal small bowel and colon in healthy animals. Should be up there in that free mucus or scattered mucus region. Now, with ongoing intestinal inflammation, we see those bacterial groups move closer to the epithelia, and they start to congregate in the adherent mucus or attaching to the epithelium or invading within the mucosa. So beneath there, I put a panel of 6 3-color fish images. So panels A, B, D, and E show you the classic biofilms that occur in these dogs with chronic enteropathies. If you go back and look at the human literature, particularly some of the early work by Swidzinski, in the 205 to 215 range, his images are identical in humans with Crohn's disease and ulcerative colitis. Then finally, panels C and F show you what we expect to see in healthy animals with these individual bacterial populations that are fluorescing orange, found predominantly in that free or scattered mucus region. So what did we find in the second study? Once again, little change in the fecal microbiome, no change in clinical score, that was a little surprising, no change in histologic score, and no change in the numbers of mucosal bacteria. And so that was hard to wrap my head around. How could we be giving, up to 400 billion organisms per day to an animal and not have significant changes to those animals that receive the probiotic? Nevertheless, what we did follow through on the IHC evaluations in the ileum or colon, we saw marked changes in tight junction protein expression. As predicted, healthy animals have high levels of the 4 tight junction proteins evaluated. At baseline, IBD dogs were depleted in all 4. And what did we see post-VSL3 administration? That 3 of the 4 tight junction proteins were significantly increased in those mucosal tissues. And this included increases in E-cadherin, occludin, and zonulin tight junction proteins. I wanted to share one recent study that we completed in our laboratory group, taking a look at the role of a hydrolyzed diet supplemented with prebiotics as well as glycosaminoglycans. And so this was a randomized clinical trial in 15 IBD dogs. They were fed the hydrolyzed diet for 10 weeks, either alone or supplemented, once again, with the mannan oligosaccharides, the MOS, or the glycosaminoglycans. And the aim or objective of this study was to do a targeted metabolomics approach using the serum in these dogs to compare the influence of these various treatments on the serum metabolome. And so what did we see? Well, we saw marked changes. And so whether the group got hydrolyzed diet or hydrolyzed diet plus supplements, we saw that there was a difference in where these dogs were at diagnosis and where they were following 10 weeks of the dietary intervention, either with or without the supplements. Importantly, when we evaluated the various serum metabolomes, there was a marked increase in lipid classes, particularly lipid classes called glycerophospholipids. And so glycerophospholipids are important structural lipid components of cell membranes. And so good examples would be sphingolipids or phosphatidylcholine. And so the take-home message from this study is, okay, using this targeted approach, we saw changes in the serum metabolome, and we saw changes in metabolomic products that are thought to play a critical role in protecting cell membranes, implying that perhaps hydrolyzed diet with these supplements would be beneficial to reduce intestinal inflammation. And so it's clear to us, as well as the human literature, it's not just who's there, but what are they doing. And so the concept of a core microbiome is not based so much on the phylogenetic members, but more so on the functional genes that are there and their metabolic products. We also know it makes sense that humans and dogs with chronic enteropathies have microbial imbalances. So intuitively, it makes sense that we're going to see disturbances in the metabolome. And indeed, our studies have shown this. Remember that probiotics may be beneficial not only because they may colonize, although many of the times they don't, but probably more importantly through the elaboration of these metabolic products as the probiotics pass through the alimentary tract. And indeed, our studies have shown that we have increased production of short-chain fatty acids following probiotic administration, and that dogs with chronic enteropathies, just like humans, have defects in select bacterial metabolites such as tryptophan or even its metabolite indole that have been documented to be low in both humans and dogs with IBD and protein-losing enteropathy. So clearly, probiotic investigations focusing on the metabolome are relevant and are clearly warranted. And our research group believes that this best information is going to be derived from metabolomic analysis. Then lastly, I wanted to show you on a recent study that we're taking a look again, focusing on the metabolome, asking questions about disturbances in bile acid metabolism as a consequence of nutrition. We've observed in our studies here, as well as elsewhere in the literature, that dogs get malignant colorectal tumors like adenocarcinomas, just like humans do. And it's also well established in the human literature that risk factor for colorectal cancer are high-fat, high-meat diets. And so we've initiated a pilot trial asking the question, if we take a group of healthy dogs, we have a service colony here of healthy animals that we can utilize for nutrition trials, and feed them a Western diet, what happens? And so we performed some preliminary studies. I've shown the example of these colonic tumors as Dr. Allenspa and I see them. That middle panel labeled A, B, and C emphasizes that dogs with chronic enteropathies, their tissues are enriched selectively with distinct bacterial groups. That panel C is a dog with an adenocarcinoma, showing these tissues are robustly enriched with Clostridialis. And then finally, our pilot studies feeding these healthy dogs a high-fat diet, not a Western diet, but a high-fat diet for 2 weeks, we see marked proliferative responses. And so it's thought the process of high-fat diets induces intestinal inflammation, cell proliferation, and alters normal cell apoptosis. And in this trial, when we looked at primary colonic tissues, we saw that as short as following 2 weeks of this high-fat diet, that proliferative indices, that is the expression of intestinal stem cell as LGR5, as well as Ki-67 is a proliferation marker, were dramatically and significantly increased in dogs as compared to a cohort of control dogs fed a kibble diet. So we think the dog is just a phenomenal translational model for investigating colorectal cancer in humans. So my last slide, special interest in technology. I was asked to let you know that I'm co-inventor of the iSurf patent that you see in front of you. And this specific patent is entitled Canine Epithelial Organoids and Methods for Making, Recovering, and Use. My laboratory group has a broad range of applied research skills. I've given you multiple examples of our expertise in clinical trials via dogs through our natural caseload that we see within the veterinary teaching hospital. Also, I've showed you the resource that we have in our service colony of healthy laboratory reared dogs that we can utilize in non-invasive, particularly nutrition studies. Spontaneous diseases in dogs translate very well to the same diseases in humans. Osteosarcoma, colorectal cancer, inflammatory bowel disease, diabetes mellitus, those are some right off the top of my head. We've established a bioarchive over the past 5 years of primary tissues and biologic specimens, as well as epithelial organoids. Finally, I have an excellent group of basic science collaborators, beginning with Drs. Wandermuller and Greg Phillips on the notobotic mouse side. The ASF mouse is a phenomenal model because it's colonized with 8 organisms that really mimic and reflect a more complex microbial consortium. And so what am I looking for? What is my research team looking for? We're looking for industry support to match NIH grant support. We have 2 applications out under review. One is a resubmission. I'm confident that one or both of those will be funded. And an industry sponsor to test our ASF model in a more mechanistic fashion. We have a couple of small studies out there pending funding, but I think we need to leverage the ASF consortium to a greater extent, using this as a platform to ask specific mechanistic questions when we can start to begin to link metabolomic production to a single bacterial species, since there are only 8. I really appreciate the opportunity to speak to you guys. And the last slide has my contact information. And more importantly than that is just an outstanding backpacking trip I went on recently to the north rim of the Grand Canyon. And thank you so much.